Our next speaker is Leandro Santos. Leandro is currently Director of Clinical Research at Insight Corporation. Leandro has worked in the pharma industry over 14 years on dermatologic products, and as a member of the expert panel on topical and transplant products, has contributed immensely towards revising this chapter. Leandro?
Thanks so much, Dr. Tucker, and good morning, good afternoon, everyone. Really pleasure to be here presenting on behalf of the expert panel of USP 1724. Before I get started, let me just make sure I put here as a laser pointer. Little disclaimer for my legal team. What I would like to discuss today is some of the key changes that Dr. Marcus has alluded in her presentation, right? So prior to me, and as we have, you probably are aware, right? So we have changed some of the layouts on the chapter 1724, as well as I think one of the major points, including IVPT, and provided some of the general points about IVRT and IVPT, right? So I think the major point that once this chapter goes out for revision or for comments from the general public, right? So we are going to notice this inclusion of in vitro permeation testing and a number of subsections, right? So some of these points, as I'm going to be showing my presentation today, may have been discussed, some are new, right? So I think the relevance here is to see this in the context of the USP, right? Additionally, you are going to notice that we try to use generic diagrams for the apparatus, right? So as you may recall previously in the USP chapter, there were some very strict measurements for the diffusion cells, for instance, that should be used. I think the trend, as you're going to notice, is to sort of move away from these very specific measurements and focus more on the scientific principles of the apparatuses. Further, we also have included some subsections on equipment maintenance and qualification. I'll touch base briefly on those in a moment. And finally, we have also done some layout changes on the IVRT section to sort of mirror it with the IVPT. Right? Moving on. I think this is the first part that everyone will be noticing once the chapter goes out. And I want to make very clear, this is still tentative section layout, right? So Dr. Marcus mentioned the time we have been working on this actually has been almost 2 years since I joined the panel. I think the panel was meeting even before I joined. And at one point I do want to highlight the scientific depth of the conversations that we have been having on a somewhat regular basis, right? So I think this is something that happens behind the scenes. Putting together a revision like this, it's a major effort, right? So coordinating across many areas and scientists, but all I can say is we have been trying to discuss a number of points, right? So it's not something that has happened, quote unquote, overnight. And the first points that you are going to see that we tried to mirror parts of IVRT and IVPT. I think this is extremely helpful for someone becoming familiar with these two techniques, right? Because the first thing that you're going to notice is if you're trying to read more about the synthetic membranes, of course, there is the chapter 1002, right? So that will be written, put together along those lines. But if you want to read more about the biological membranes, you also have some intro there, the more common types of equipments, analytical methods. Of course, there are differences in what you're going to use for IVRT and IVPT. In terms of experimental design, I think this is extremely helpful because it helps understand the nuances, the differences between these two techniques. They're extremely important in topical development. And finally, some other sections about the use of IVRT in scale-up post-approval changes, and some of the comparisons of IVRT versus IVPT. In this case, I'll be showing later in this presentation how this looks like. Okay. The first point that I do want to say, I mentioned about the tables that we have been trying to do these comparisons. The intent here of this slide, of course, is not to go through each of the comparisons. However, I think it's particularly interesting to contrast IVRT and IVPT. It's not uncommon to have some confusion when to use each of the techniques. I think for those who have been attending the workshop since yesterday, we have had two extremely informative presentations by Dr. Rainey on IVPT yesterday and IVRT today, right? So this is a great resource for anyone in the future referring to these presentations. And beyond that, if you, for anyone looking at the 1724 chapter and trying to contrast these two techniques, you can see some of the clear differences, right? So IVRT, the focus in here is to evaluate sameness of formulations to understand drug release of a reference product versus a comparator product. Right? So, and as you can see on some of these points that we have, compared batch to batch variability, definitely it's something, for instance, you are not going to use in vitro permeation testing, given the intrinsic variability of using human skin, which is the case for IVPT. On the other hand, if you focus on the upper part of this diagram that we discuss when IVPT can or should be used, for instance, in cases, let's say if you're an innovator, you're trying to select new chemical entities, NCs for further development, or if you're trying to optimize new formulations, primarily you want to use IVPT to understand the drug distribution across layers and all the flux. Of course, at a later stage, you're also going to use IVRT, but the idea is to contrast when you are going to use one technique versus another. Right? So I think this is extremely helpful and a sort of a quick way to differentiate the two techniques. Now, as we go through further, right, so on these comparisons, you probably have seen a somewhat similar table earlier today. And again, intent here is not to go through every single detail, but those are the key points, as you can have here, the key parameters whenever we are comparing the two techniques, right? So major differences in terms of IVRT and IVPT. IVRT, we're talking about synthetic membranes. Of course, there's a lot of science on how to select those. And then like VPT, we are focused on biological membranes, primarily focused on a human skin, ex vivo or cadaver skin, as was extensively discussed yesterday. Receptor solution, extremely important component as well. Typically, IVRT is somewhat common to have aqueous organic combinations, sometimes ethanol, IPA, acetonitrile. On the other hand, IVPT, you want physiological conditions, right? So you're generally not going to vary much from phosphate buffers, saline, and sometimes you may need to have some additives just to ensure sink conditions, as you have discussed. Sampling, there are some differences here and there. In terms of the apparatus, as you can see, primarily vertical diffusion cells, sometimes immersion cells. We also have used apparatus for that has been discussed in the past, right? So, and I think there's an important conversation still ongoing on the apparatus that will be including and discussing further in this revision. For IVPT, VDCs, of course, still well used and very much discussed during this workshop. And now the flow through diffusion cells, not to confuse that sometimes the apparatus 4 is also, right, so mentioned as flow through diffusion cells. Here, I'll show the diagram in a coming slide, but just to mention that difference in terms of nomenclature, and that's what you're going to see on the revised chapter. We tend to use this term FDC and VDC, right? So just to make that differentiation. And overall, some other aspects that I'll probably be discussing in the next two or three slides, but the differences in terms of the amounts in the receptor solution that obviously is going to affect the analytical method of choice. Generally speaking, you are fine using LC, HPLC with UV detection for IVRT, right? So the matrix or the samples are fairly simple. The concentrations tend to be large, microgram to milligram, as you can see here, as opposed to IVPT, right? So it's a different story. Now, more and more people have been using mass spectrometric detection. Some can still use LC with UV detection, assuming your compound is amenable to that type of analytical technique, and how you're going to display your data, right? So we still have some presentations today and tomorrow to discuss how to present your data, how to discuss, but of course there is a difference given what you are looking for in each one of these techniques. IVRT, mostly your release rate, your slope, as opposed to IVPT that you're looking your flux profile, your Jmax, and the cumulative amount, IMT, as was discussed yesterday. As I mentioned, right? So here I have some of the generic diagrams for the apparatus we have. So if this is still, as I discussed, as part of our ongoing conversations within the expert panel. But the idea here, once again, instead of having exact measurements for each one of the apparatus, we want to focus on the scientific principles. And this is something has been mentioned over and over again, right? So understand your equipment well, right? So rather than being restricted to specific measurements or a specific vendor, whenever we try to understand qualification, whenever we try to understand the critical parameters of your method development, it's critical that you do understand the equipment that you have in your laboratory. Right? So here we have the usual vertical diffusion cells. You have seen this a number of times. As I mentioned, the flow diffusion cells, the FDC, right? So not the USP apparatus 4, and just a schematic of an immersion cell. So the summary that I have in this slide is, once again, we're trying to move away from very specific measurements, instead trying to focus on the scientific principles of the techniques, IVRT and IVPT, and how do you choose your apparatus in that context. Now, talking about IVRT, I'm mindful that some of these, or most of these parameters, they have been presented or discussed, right? So I don't want to rehash everything else, but the general points, as Dr. Marcus alluded, for membrane selection, we're going to have the separate chapter 1002, right? So I think it will be extremely informative to help us define, select, how we're going to evaluate if a membrane is adequate given the analyte you have in mind, given the receptor solution that you're going to use for your VRT method. In terms of the diffusion cells, mostly, and you are going to notice that I tend to use typical, right? So there might be other, diffusion cells or maybe other apparatus that can be used, but here we tend to focus on what is typical. Also, in terms of analytical method, I discussed that. And here, I think some of the key points that had been discussed or presented this morning by Dr. Rainey, and I'm sure will be a number of discussions this afternoon along those lines, and even in our panel soon after my presentation. But the idea, generally, you are looking for a pseudo-infinite dose occluded. There's some details on the application methodology, right? So we do not want to apply using some sort of device that can affect the microstructure of the formulation, that can have an effect. This was discussed yesterday for IVPT. The concept applies to IVRT as well. The receptor solution, generally, we're going to have aqueous organic, right? So I think extremely important to ensure that whichever you choose in terms of receptor solution is not going to diffuse back into your donor compartment where you have your formulation, and then is going to redissolve that formulation and, of course, affect their composition, right? And important considerations during method development. Experimental length, typical, again, in focusing on that word, equal or less than 6 hours that we have seen. Remember that you want to properly characterize, right? So your the release profile of your formulation, and generally what we have seen is equal or greater than 5 sampling points. Number of replicates, generally you are comparing 6 per formulation, right? So if you have your reference product, you're going to have 6, and then your comparator product, you're going to have 6. Like, so generally you're going to run 12 cells. Some of these experiments you may need more. Purpose here is not to discuss all variables that you're going to take into consideration. Membrane temperature, If you're talking about a product that's intended to be applied on the skin, generally they're looking at a 32 plus minus 1 Celsius membrane surface temperature. Recall that you need to understand your equipment well, so you control the temperature and ensure that the surface of the membrane is 32 plus minus 1. If you're talking about some products, for instance, products for a vaginal application, then you need to consider a different temperature. Now we're talking about 37 plus minus 1, and that's the reason we have here internal. And as I discussed, data reporting tends to be release rate. Of course, the chapter it carries over some of the information that has been already used in the version from 2013, right? So we're not changing that as much. We still have all the calculations there. Some additional points, just and I want to be clear, those are areas we're still discussing the upcoming meetings with the expert panel, but to expand or revise some language around sampling procedure, how to ensure your receptor solution temperature is controlled, system equilibration, right? So a very relevant, as has been discussed in some of the presentations, that before you start the experiment, you are sure that everything is good to go. Your system is equilibrated. You are not going to have anything else that's going to affect the data quality. So shifting gears, I do want to show something similar, although substantially more complex, around IVPT. And very mindful that some of these points have been discussed many times since yesterday, right? So I think everyone might be a little bit tired of seeing this, but what we're going to see in the chapter, again, the focus is on a human skin, dermatome, generally speaking, is what we tend to use. We have discussed already in this workshop some of the challenges as we start, for instance, trying to separate stratum corneum or trying to separate epidermal sheets. You start doing a lot of manipulation of the skin that may actually include errors or in your sample prep. Generally speaking, what we're focusing this chapter is human skin is dermatomed. And being consistent in the anatomical area, right? So if you start developing your method with trunk skin or abdominal skin, you should continue with it. You should not be switching, starting with facial skin, let's say, very extreme example, and then switch, for instance, to trunk skin or abdominal skin. So be consistent once you do that selection during your method development and as you go through in your pilot study as well as your pivotal studies. Diffusion cells, again, what is typical, right? So VDCs and FDCs, flow diffusion cells, those are the ones you are going to see more commonly in the context of IVPT. Bioanalytical methods, validation is encouraged. We have discussed this a number of times. Extremely important that you know that your analytical method is going to work as you expect it to, right? So you can run, again, a perfect IVPT method. If your bioanalytical method is not good enough, your data quality may suffer, right? So you may not have reliable data. And the other points, I just want to say, like at a high level, the differences, right? So now in IVPT, we're talking about finite dosing. Again, typically, we had a very lengthy discussion yesterday about dose selection, but generally speaking, most of the methods, they're going to focus at equal or less than 15 milligrams per square centimeter, unoccluded. I mentioned again the application methodology and the relevance of it, so I won't repeat myself. Receptor solution here, you are looking for something that's more similar to physiological conditions, right? So now you take a very different approach from what you do in IVRT. You do not want to affect the skin barrier. Therefore, in IVPT, you do not want to be adding ethanol, isopropanol, because that's very likely going to affect the skin barrier, right? So that's something you do not want to have in IVRT. In terms of experimental length, typical 12 to 72 hours, 8 non-zero time points, and this is generally the minimum, right? So you want to have 4 non-zero time points prior to your J max, at least, right, 4, and then at least 4 non-zero time points after your Jmax. So this is a general recommendation. You may need more than that. Same goes for donors, right? So donors and replicates, we had a discussion, if generally speaking, if your method has less than 4 donors and 4 replicates per donor per treatment, you may not be able to generate meaningful data that helps you make scientific decisions. The surface of the human skin in this case, or the biological membrane surface, 32 Celsius. And what I do want to say that in the context of IVPT, we are primarily focused in this chapter on human skin. We are mindful there some other biological membranes or some other epithelial membranes that can be used, but as you read this chapter, once it becomes available publicly, you are going to notice that. Membrane integrity, focus. Yesterday, we talked about transepidermal water loss. There's some other methodologies. I think the intent here is to potentially expand and have a separate chapter that could discuss this aspect of membrane integrity, right? So it's something not trivial, but again, the trend or most of the most recent papers, they tend to look at transepidermal water loss to assess membrane integrity. Drug titration distribution and mass balance. This is typically not done for some of the generic applications, right? So we talked about that yesterday, but in the context of the innovator, you may want to understand mass balance. You may want to understand drug distribution into the epidermis and dermis. So we have some lines, or I think 2 or 3 paragraphs talking about this part. And then data reporting. As I mentioned, we are looking to have Jmax, your flux, and your cumulative amount, AMT, as we want to start using from now on. I know that was a lot of information. So, but this is actually my last slide, okay? And the equipment maintenance and qualification section of 1724 or of the revised 1724, I would say this is still in flow, right? So we have a number of discussions and edits, but the general nature of that subsection is try to provide some guidance, right? So as you can see here, suggestions, right? So of when to do your requalification every 6 to 12 months. But a number of important aspects. Once again, make sure you understand your equipment. Make sure you understand what are the areas that are more prone, to issues, right? We talked about, and Dr. Rainey actually mentioned about these, understand your cell size and volume. Do not use nominal values. It's very important that you measure the volume in your lab, and then you have that for each of the cells. Some of the cells, they are made of glass, right? So they tend to have less variability. But when you talk about other components, right? So let's say if we talk about flow through diffusion cells, tubing length is very important that you understand the void volume there, that they have the same length. And some of these tubings, remember, they are made of polymers, and it's something that I consider a movable part, right? Because you may be stretching them as you connect to the cells. So these may need to be requalified more frequently than something like a diffusion cell that's made of glass that probably is not going to really undergo a lot of changes, right? So again, understand the equipment you have. In terms of heating systems, and this applies, right? So VDCs and FDCs, there are a different number of heating systems that can be used, right? So the idea is you want to have thermoregulated diffusion cells. Sometimes people tend to use water baths. Sometimes there is dry heat. But the main point here is to make sure that you have something that's thermoregulated, that's not going to vary, right? So that's within the specifications of your system. Steering devices also got to understand that this will affect, of course, your sink conditions. And if you don't have good mixing, this will also affect the concentration of your drug as you sample it. Right? So make sure that this is understood. In the context of the flow through diffusion cells, now we're talking about the flow rate, right? So you tend to use some pumps, right? So that are going to help that. These also need to be qualified every so often. And overall, what I would say, right? So for some the flow through diffusion cells, it's a setup that tends to be, I would say, slightly more complex, right? So again, if you are going to use FDCs, you also need to consider tubing alignment for proper sample collection. As I said, replacement during regular intervals, especially if you see that the polymer is getting like yellowish or is getting over time oxidized, right? So this is not something uncommon to see, but just make sure that you are paying attention to those aspects of the systems. Here, just a small observation in terms of immersion cells. Something, honestly, I have not worked hands-on yet, but we have other members of the expert panel who have had experience with these. So I just added a comment that these tend to be more uniform in terms of manufacturing. So generally, the variability here is lesser, right? With that, I'm actually going to hand back to Dr. Tucker so we can go to the panel discussion. Thank you very much for your attention today.